Since India's independence, many researchers have made major contributions to the world of science. Written Out of History touches upon the lives of 3 forgotten scientists whose work has redefined biological research. Infectious diseases have long haunted humans. The most recent COVID-19 pandemic has brought the entire world to a halt. But there's another such disease more than 200 years old. It still continues to infect 4 million people each year. Cholera. A discovery made by a scientist in a lab in Calcutta changed the way the world understands intestinal and diarrheal diseases. Yet, very few remember him. Professor Sambhu Nath.
আমরা দেখতাম বাবা রিসার্চ করছে সেই রাত্রির বেলা মেডিকেল কলেজ চলে গেল রাত 10টা কি অত কিছু একটা দিয়ে করেছে তার রিয়াকশনটা দেখতে গেল এই গেল মাঝে মাঝে এরকম চলে যেত
We saw his coming to the lab, our lab, by driving his own car and carrying a cold bucket which contained the Vibrio cholerae culture. Sometimes we used to say, the Dr. Babu, you are spreading cholera in our lab. And he used to reply jokingly, no, Baba, no. These are all killed cultures. So I'm cleaning everything. There is no fear of cholera.
Calcutta in 1949. In the aftermath of partition, the city was teeming with refugees and the hospitals with cholera patients. At the same time, with a PhD in hand, Sambhu Nath returned from London. He was appointed the head of pathology at the NRS Medical College, where countless cholera patients were admitted. As someone who grew up in a village near Chandannagar in Bengal, he was all too familiar with the harsh realities of life in rural India.
He has seen probably cholera patient dying in front of him, and that really drove him to work in this area. It's a passion to serve the people and community at large.
Professor Day began studying the pathogenesis of cholera, looking at how its bacteria affects the human body. But where did this disease begin?
Cholera is an ancient disease. In 1817, the first cholera case was reported, and it was thought to be emerged from Gangetic belt. And that time it was confined to Indian subcontinent.
Gravely remembered as the Blue Plague, the world has observed 7 cholera pandemics until 1975, which have caused more than 38 million deaths in India alone.
So people get infection of cholera by ingesting either contaminated food or water. Orally, it enters into the system. Then it reaches the stomach. There it bypasses the gastrointestinal barrier, and then it reaches the intestine, where the pathogenesis of cholera starts.
The cholera bacteria was discovered in 1884 by a famous German physician, Robert Koch. He also hypothesized that an endotoxin present inside the bacteria caused diarrhea and vomiting.
This toxin can cause huge outpouring of fluid in the intestine and then purging of the fluid through stool, causing severe dehydration, which is responsible for death in case of cholera cases until it is treated immediately.
Koch assumed that the bacteria affected the circulatory system of the body, and most scientists didn't really look elsewhere. Professor Day was able to look over Koch's assumptions. He presumed that the bacteria targeted cells lining the small intestine. To prove his hypothesis, he developed an animal model.
He did a very simple experiment that time, because that time there was no advancement in technology, even very limited resource and no funding. he took the culture filtrate of the Vibrio cholerae and he inject them in a segment of intestinal loop in rabbit model and ligate the both end of that part with silk ligature. After 24 hours, he saw that the intestinal loop was full of secretions.
These secretions were analyzed at the Bose Institute with the help of Professor Day's friend, Mr. A.K. Sen. Working in a different institute as a guest scientist without any red tape is now a thing of the past. Professor Day had a feeling that a toxin inside the bacteria could be responsible for the secretions and not the bacteria itself. And to test his theory, he repeated an experiment multiple times by injecting just the toxin without the bacteria. He was right. He saw the same results every single time.
Actually, the exotoxin, not the bacteria, is causing the cholera. And this exotoxin acts on the intestinal epithelium. It produces secretions and there is a huge loss of water and salts from body.
It was of huge importance because before that, people searched all over the body for the toxin. But he was the first person who could detect if you put toxin in the intestine, you get the cholera. So that is the beauty of the method.
ফার্স্ট নেচার পত্রিকায় বেরিয়েছিল এবং নেচার পত্রিকাটা হলো একটা সায়েন্টিস্টদের বিরাট সম্মানের জায়গা, সবাইকার বেরোয় না কাজ, ওটা নেচারে ফার্স্ট বেরিয়েছে।
Professor Day's work was hugely significant for pathology and public health worldwide. His observations pointed out that loss of fluid was the leading cause for death in cholera patients, and by replenishing body fluid, many lives could be saved. His work paved the way for oral rehydration therapy.
The mortality of cholera in ancient ages, it was 50% without treatment, which has come down to less than 1%. So we can understand the importance of this discovery by which the oral rehydration therapy has been established.
এটা একটা বেসিক ডিসকভারি, এটা কোনো কারোর থেকে না, একটা নতুন মানে নোবেল প্রাইজের জন্য যে ইয়েগুলো দরকার সবগুলো তো ওতে ছিল না বাবার কাজে। না চাইলে তো আমি বলতাম তোমার তো এটা তো মানে 100% তোমার পাওয়ার।
His name was 2 or 3 times recommended for Nobel Prize by none other than the another Nobel laureate, Joshua Lederberg. But again, Dr. Day at that time, he was introvert in nature. He was reluctant to give a publicity for it because he used to say, okay, if the work is good, people will come and ask for my data. So I don't need to publish.
Even I am his grandson. I came to know about his work when I was doing my MBBS. And that time I first realized the importance of his work. I read it in my microbiology book, that the Day's loop method, which is universally accepted.
Just as we are witnessing newer strains of coronavirus emerge because of rapid mutations, cholera also underwent one in the 1960s. The classical strain of cholera was replaced by a weaker L-tor strain, making the disease less lethal. The world could now start witnessing the end of cholera, but the importance of Professor Day's science was going to lie uncelebrated in the pages of medical textbooks.
He was really disheartened because his whole technique was based on the classical cholera. He used to lament, my old classical cholera is gone. And that is one of the frustrating things which probably prompted him to give up his research later on.
রিটায়ার করলো, তখন ভাবলো যে অ্যাক্টিভ নিজেকে রাখতে হবে, তখন ছোট করে ল্যাবটা করলো। আমি বাবার সঙ্গে একমাত্র আমি তো থাকতাম, কাজে আমি বাবার সঙ্গে ওই ল্যাবরেটরিতে হেল্প করতাম, কাজ করতাম দুজনে মিলে। হ্যাঁ, একটা আপনাকে ওটা দেখানো, ওটা রয়েছে, একটা ইজি চেয়ার ছিল, এইখানে থাকতো ইজি, ওইটা হলো বাবার। ওই সন্ধে হলে না বাবার ইজি চেয়ারের মাথা দিয়ে কী চিন্তা করতো, ওই সন্ধেবেলাটা না একটা কী হতো সাইকোলজিক্যালি, বাবা ভীষণ ডিপ্রেসড হয়ে। During his last days, he was disappointed and disheartened, no recognition, no honor. Though he was not like that to pursue the monetary or that way, but still a scientist need recognition at least.
When people came to know of his work, they asked, who was Dr. De? How come we don't know his contribution? Did he get any award from the government of India? Padma Shri, Padma Vibhushan? Find the list. No, he's not there. Why we missed him? Why on earth we missed him?
Finally, in 1978, he was invited to attend the 43rd Nobel Symposium on Cholera and related diarrheas to express his views, where he said,
I have been dead since the early 1960s. I have been exhumed by the Nobel Symposium Committee. And these 2 days with you make me feel that I am coming to life again.
He used to do all the work with his own hand, cleaning everything. And he was handling a lot of patients' blood. During the handling process, he got infected with hepatitis. At that time, there was no vaccine for hepatitis. I remember my professor, when he went to visit him in his deathbed, then Professor Sen asked, how did you contract the disease? Dr. De painfully smiled and said, professional hazard.
it took almost 50 years between Koch's identification of the cholera bacteria and the discovery of the exotoxin that causes the disease.
Because of this discovery, the pathophysiology of various intestinal diseases and diarrheal diseases caused not only by Vibrio cholerae but by other enteric organisms could be understood.
We know a lot of things, particularly after the discovery of Dr. De on Choleratoxin. But again, control of cholera not only depends on the scientific research, but also it has economic annotations.
Today, cholera is a disease of the developing world. Lack of sanitized potable water and a hygienic sewer system leads to cholera outbreaks. And economic disparity directly equates to the number of deaths caused by it.
We get still epidemics of cholera here and there. India and Bangladesh used to be the home of cholera, but now it has spread to many other countries, including in Africa, where it was not known to be of prevalence.
Communicable diseases like cholera have been a huge health burden for highly populated countries like India, where resources are scarce. The COVID-19 pandemic has emphasized the need for stronger public health systems, and a timely recognition of scientists like Professor Sambhu Nath can inspire many more researchers to work on local problems.
I decided to study botany because it was my favorite subject in school. I was awed by the contribution of Sir Jagdish Chandra Bose and fascinated by his work that showed that plants were living organisms and had a metabolism similar to that of animals. I developed a strong determination to find the locations of the heart and brain of plants and to understand the way they functioned.
I had once seen a lady, coming from the special university buses and coming down. She was wearing a long coat, very elegant. And I always thought what kind of a person she would be, because he would always say she's very dynamic, very enthusiastic. And I really wanted to meet her someday.
In the 1960s, before the advent of genetic engineering, Professor Sipra Guha Mukherjee was an embryologist at Delhi University. She worked in the unit established by Professor P. Maheshwari, the eminent botanist who pioneered the classification of plants and application of embryology in India.
I first met Shipra somewhere in early 70s. That time I was doing PhD in Delhi University. So Maheshwari was interested in cell division and cell cycle processes. He had asked her to use anther culture as a system to understand how the cell divisions occurs in the microspores.
Pollen grains, also called microspores, are the male sex cells of a plant found inside the anthers. And anthers are the male reproductive part of the plant. These cells go through a special kind of reproduction called meiosis, where each cell divides twice to produce 4 cells. She was trying to understand the biochemistry of meiosis, something that wasn't well known at the time. She was working on anther cultures in the datura plant, which is known for its large anthers. During one of her experiments, Professor Mukherjee accidentally discovered something that would change the way we produce lab-grown plants.
Plants have a very great trait. It's called potency. Any part of the plant is capable of regenerating into an entire plant. You take a cell, you take a part of a tissue, a root, and they will regenerate into an entire plant. This particular feature was first discovered in 1950s. Then came Dr. Sipra Guha Mukherjee's momentous discovery.
So they had cultured anther and they were trying to study the effect of cytokinins. These are growth promoting hormones. They added coconut water, which is very rich in that, and they just left the cultures. After some time, Sipra Mukherjee, she noted that there were some protrusions coming out from that.
This was an excitement which she talked to Professor Maheshwari that what this could be, and they found that these were actually some kind of embryos which were emerging out of the anther.
She was going to throw away her cultures. She thought her experiment was over, and then accidentally she discovered a haploid organism can develop into an entire plant.
Some of the world's most important discoveries have been accidental. Everything from penicillin to X-rays and LSD to Viagra. Professor Mukherjee's accidental discovery paved the way to producing haploid plants, a plant that can grow just from pollen instead of the usual fertilization of eggs and sperms to form seeds.
It's something like saying from sperms one can make a human being. So it's that radical thought.
People had been trying since 1928 to develop haploids, not through agriculture, but haploid has a lot of usage in agriculture. But this turned out to be the most interesting, simple method, and you can get haploid in large numbers.
The implications of this discovery were huge. Haploid plants have one set of chromosomes and are capable of carrying a desired trait. These can be converted into diploids to reproduce the same desired trait in future plants.
So that is the main turning point, that it provided a very efficient tool for plant breeding by giving you a hold on to the trait that you want to.
Professor Mukherjee was interested in making a real impact with her science. So she turned her attention to rice, because over half the world's population eats rice daily. She was successful in using anther culture to produce haploid plants of rice.
India actually, after this discovery, there were not too many groups which came up. In fact, there were some groups which were even doubting whether these are really haploid in nature. And she later on went to Indian Agriculture Research Institute, worked also with Swaminathan, and demonstrated that not only in Datura inoxia, she showed it also in rice, that rice can also make the haploid plants.
Sorry to say, at that particular time, nothing was done that this particular discovery could actually be used for applied purposes in agriculture. Whereas the other countries like China, they went far ahead. So as far as rice was concerned, normally you have 2 seasons, but they could have 3 seasons. They could get high-yielding varieties. They could get pest and disease-resistant varieties. So all by using haploids.
For Professor Mukherjee, the challenges went beyond just the acceptance of her discovery. Most women in India at the time couldn't dream of a career in science.
I felt I was swimming against the tide. No woman scientist could rise above a certain level, and thus we felt mentally inferior to male scientists. Even in the US, the situation was not very different.
At that time, there were no women, literally no women who were faculties at the university. So she was one of them who fought for that and told Professor P. Maheshwari he should be, also giving chance to the women to accept such positions. And he blatantly said no because, they will get married, they will get pregnant, and they will not come back.
She had to fight the world, which was dominated by male scientists, but she would not fear anybody. She just went past and she actually made her name. And she encouraged us all to go ahead and, pursue science as a career.
While doing her postdoc work in the US, she was invited to take up a faculty position at the newly established School of Life Sciences at the JNU. Professor Mukherjee would become the first woman faculty of Jawaharlal Nehru University.
Shipra Mukherjee was the pioneer in setting up the School of Life Sciences. She came from abroad. She had fantastic ideas, what kind of course curriculum should be there, tutorials, and all kinds of things.
Professor Mukherjee's personal life was as revolutionary as her professional life. She chose to return to India from the US after her marriage to mathematician Narayan Mukherjee, and it took 7 years for them to be reunited.
Every single day at lunchtime, one letter to her husband. That is what she used to write because they were apart for some time.
Our father, the late Professor Narayan Prasad Mukherjee, his life as a mathematician at that time would have been certainly better had he stayed in the United States. He chose to come back to India just to be with my mom and support her.
On the professional front, her work started receiving international recognition. She was invited to present at the EMBO Symposium in Italy in 1971 and the following year in Canada. But it was in China that she received the most gratitude.
So she was really received in China as somebody in the scientific field who has achieved a mark in her career.
I met Professor Hu Han, who appreciated our work on anther culture and remarked that our work was responsible for a substantial change in the agricultural economy in China. This was undoubtedly one of the greatest moments in my life.
Professor Mukherjee's personal and professional life faced a massive setback in 2005.
She had a tumor in her right kidney in 2005 and she never realized it. But then the cancer metastasized in her brain and that was terminal. And I'm sure she went through a very, very terrifying time because even the doctors told us that she was going through a very, very hard illness. But not once did I see her break down. Not once did I see her cry in front of us. Towards the end, she shut out the world because I think it was difficult for her to open her eyes and communicate.
And then June she had it, then July, August, September, around 15th September, she passed away. The amount that I cried, I think they were actually shocked because she was part and parcel of my life and I couldn't imagine a person like just going away, and she was not that old. I think maybe she was 67, 68.
We used to teach many courses together. Many of our research projects were together. And she was a wonderful person. So we'll remember Shipra for her contributions, for what she did to the university, to School of Life Sciences.
She taught us the small things of life which, other people would not even bother because she was herself a perfectionist. And there's a big vacuum, plants. And we all remember whenever we say, oh, Shipra jaise koi nahi tha.
She helped develop careers of many other women and other faculty members. And she got students who also did very well. I think that was a good part of her life that she accomplished.
Professor Mukherjee's legacy continues to shape the future of plant breeding in the world, and it remains one of the crucial tools for food safety. Arundhati Mukhopadhyay is part of a team of scientists in Delhi University using the principles of Professor Mukherjee's discovery. She wants to improve the quality of Indian mustard to better suit the needs of India's growing population.
Indian mustard has very sharp taste and it has also very strong smell. Many people do not like it. So to remove this by conventional breeding method will take years, like maybe 30 years or so. And we have to always keep in mind while doing this, the oil production should not be compromised. So by conventional method, this will take about 30 years down the line. But if we use this double haploid technique, it might be accomplished in 10 years or 12 years time.
If we had actually focused on Professor Sipra Mukherjee's discovery at that time, today with this changing climate, we would have had more resilient plant and crop varieties which could tolerate drought and heat conditions.
Indian science has come a long way from the times of Professor Sipra Guha Mukherjee, but still has a ways to go in acknowledging the contributions of its women scientists.
Today, when I look back, I have no regrets, and if at all I could change anything, I would certainly like to also understand the psyche of many of my contemporary scientists and administrators and their attitude towards women. It is important to eliminate the damage caused by such attitudes so that in the coming generations, women scientists will not have to waste their time combating them.
What can a fruit fly teach us about human behavior? Why do we feel nostalgic when we smell the rain? What do our genes tell us about diseases? Asking these fundamental questions was an exemplary biologist, one who would eventually become one of the foremost institution builders of our time, Professor Obaid Siddiqui.
When we have stories of scientists, from the very beginning, they were very attracted to it. But here was this man who was not very attracted to science. He didn't think that this is going to be his career. He didn't think very seriously about it.
It was while studying plant genetics at the Aligarh Muslim University and later conducting research at the Indian Agricultural Research Institute that his interest as a scientist was sparked.
He was looking at the wheat genomes. There was a hailstorm and he told me that the whole field was gone and he was in a fix what to do. People suggested go back home, you can repeat the experiment there, but he was thinking hard.
He wrote to Professor Pontecarvo, a geneticist at the University of Glasgow, and on his insistence went on to do a PhD in the gene structure of the Aspergillus fungi. Then came his first major discovery while working with Alan Garren, a geneticist in the US.
There was a great tide rising in molecular biology and genetics. These were all people who were right at the center of discovery, as was Obaid himself.
Obaid's work with Alan Garren led to the path-breaking discovery of a special genetic mutation in the DNA that signals the early stop of protein production. This was just the beginning of a legacy of many discoveries and of defining institutional science, but it wasn't easy.
Obaid wasn't sure about where he wanted to go back to India, and he wrote to Homi Bhabha about joining TIFR. That was the beginning of the molecular biology unit. It had nothing there, and it was a very, very difficult time. But he, right from the beginning, talked to the leadership directly and got things done. That connect, that daring to get things done, allowed the molecular biology group to survive in a context where it seemed very alien.
His success with institutions had begun, but he did not give up his own research. He went on a sabbatical with Professor Seymour Benzer at the California Institute of Technology. Together, they studied how mutant genes affected behavior in the Drosophila melanogaster, or the fruit fly, which was their chosen model animal.
All life has a common origin and therefore a common chemistry, and that chemistry is connected by the thread of DNA. By studying any organism, you learn a lot about any other organism.
70% of human disease genes are actually represented in flies, and 60% of all genes are represented in flies. So it makes a good model organism to study cell signaling or diseases or different kind of genetic pathways in flies.
He worked with temperature sensitive mutants. So from that work, he made many discoveries related to neuronal signaling and the synaptic transmission. And he decided to focus on sense of smell and in general neuroscience.
The sense of smell or olfaction is an important evolutionary sense. Memories, learning, and ultimately behavior is affected by the odors that we perceive.
He was very much interested to understand different stages of learning and memory, and he was using olfactory learning and memory as one tool to understand it. In his lab, screens were done to find out the genes that are responsible for, first of all, olfaction itself. Following that, if the flies can be trained to learn attraction or repulsion to different odorants.
The experiment involves a tea maze in which a T-shaped glass tube contains a substance with an odor on one side and a non-smelling substance on the other. Then the flies were placed at the bottom end of the T-shaped glass tube. An ordinary fly without a mutation and preferring the smell would flock to the substance with the odor. Obaid and his team repeated these experiments with several sets of flies that carry a known genetic mutation. When the mutant flies do not show a preference, it means they are incapable of smelling.
So he described 4 different genes that are involved in the sense of smell in Drosophila. So by shutting down the activity of these 4 genes, he could observe the changes at the behavioral level towards the attractants as well as the repellents.
Around that time, Veronica Rodriguez was an undergraduate in Dublin, and so she ended up working with Obaid, and she really pioneered the development of chemosensory behavioral genetics in Obaid's lab. And that put Obaid's work in a really phenomenal trajectory, giving rise to an entire new field, which Obaid did. Even though bulk of the work in the field came later on, Obaid's pioneering role was established.
In the 1990s, he helped set up the National Center for Biological Sciences in Bangalore, Karnataka. Obaid's work with the genetics of fruit fly attracted some of the brightest minds. Together, they nurtured science in an otherwise barren area.
After hiring the first batch of faculty, he really mentored all the faculty and he made sure that they had good scientific atmosphere to excel in their fields. His passion towards the experiments that we performed in the laboratory and his curiosity to see the results, all these factors really influenced me.
We were young and we were interested to publish and we were interested to finish our degrees. He said that what interests you, doing science or just getting a degree? Because if you want degree, you can get 2 of them for your work. But science is more important.
Obaid could get very strongly opinionated, and this could lead to a very ferocious discussion, ending up in people fuming and going home. The next day, it's all completely forgotten, and that happened only because of Obaid. That generosity of spirit instilled a culture where you could actually question on anything. And that's really important for science.
This intellectual culture of questioning continues to thrive in Obaid's associates and students, who are now on their own journey in the vast fields of molecular biology and neuroscience. Recently, the biology department at IISER Pune has been applying the principles of Obaid's work on the sense of smell to the problems brought on by the COVID-19 pandemic.
All of us know by now that COVID-19 patients have olfactory deficits. We have generated an animal model, a mouse model, to study the olfactory deficits that we observe in COVID-19 patients. The virus entry is mediated by the ACE2 receptors and the TMPRSS2. One of the ways to generate the animal model is knocking out this receptor type in the mouse that will mimic the COVID infection conditions. Our finding prove that we can really use the olfactory fitness to identify the asymptomatic COVID-19 carriers.
Though Professor Obaid received numerous awards for his scientific research, it was his passion for institution building that he is most remembered for.
He had this very nurturing role for scientists. The time when he was the director of the institute, he nurtured them, he nurtured their science, gave suggestions to them. For him, this was more interesting rather than the recognition part of it.
He said that he follows the philosophy of Homi Bhabha. Have people who are actually driven, who are motivated enough to do a certain thing, and then build an institution for them. This is something which can actually, if practiced, can change a lot of foundational thought about the institutions that we build today.
Scientists like Obaid Siddiqui, Sambhu Nath or Sipra Guha Mukherjee may have been left behind in the larger public imagination, but their contributions still shape the future of science in elemental ways. When our scientists do not get timely recognition, we lose out on an inspiration that could potentially define future scientific pursuits. It's time we change that.